Forests are used for many recreational purposes, like hiking, taking a picnic, and studying nature. However, being in the forest does not come without risk. Ticks, parasitic arachnids that are abundant in most temperate forests. Sucking blood, they parasitize a number of mammals and birds during their life cycle. These include mice, deer, and domesticated animals, but also humans. During their life cycle, ticks can obtain the bacterium Borrelia, which causes Lyme disease. The first sign of Lyme disease is erythema migrans, a red ring around the tick bite. Untreated, Lyme disease can in some cases lead to death. The abundance of ticks has increased in recent years. In order to ensure the safety of recurrence, understanding the behavior of ticks is of vital importance. In this study, the abundance of ticks in different forest types is investigated in the field. In the lab, the behavior of ticks is observed under different light and humidity conditions. In order to find a host, the tick climbs to a higher position and spreads its front legs. This behavior is called questing, but it is costly in terms of respiration. To restore its water content, the tick needs to alternate periods of questing with periods of quiescence in the shady, moist litter layer below. Our research question is formulated as follows. What is the influence of different microclimatic factors on the behavior of the sheep tick? This research question is divided into two sub-questions. How do vegetation structure and litter layer affect the abundance of the sheep tick? And what is the effect of relative humidity and light intensity and their interaction on questing behavior of the sheep tick? The first question is answered using data collected in the field, while the second question is answered using lab experiments. It has been observed that the abundance of ticks is highly variable among different forest sites. Our hypothesis is that this variety can be explained by several mechanisms. First of all, vegetation cover reduces the light intensity and provides shelter for host species. Therefore, we expect a higher number of ticks in sites with dense vegetation in all vegetation layers. Secondly, a thick litter layer provides shelter for molting ticks and eggs, and provides a humid environment for ticks to restore their water content. As a result, more ticks are expected in sites with a thick litter layer. Through a vegetation cover and litter layer, the abiotic conditions of the site, such as light intensity and humidity, are influenced. In the lab, the effect of these abiotic conditions on the questing behavior of ticks is investigated. Our hypothesis is that a darker environment reduces respiration and increases the chance of encountering a host, since host species are more active in dawn and dusk. We thus expect more questing ticks in a dark environment. In humid conditions, the ticks lose less water through respiration. Thus, less time has to be invested in finding water. Subsequently, more ticks should be found questing in a humid environment. Lastly, we expect that a combination of low humidity and high light conditions lead to even less questing activity, while a high humidity and dark conditions increase the number of questing of ticks. Combining wet conditions with low light conditions are expected to contain more questing ticks than dry conditions with high light conditions, as water availability is of vital importance to the ticks. We sampled 6 sites at 2 locations near Wageningen, the Grebbeberg and the Oostereng. At the Grebbeberg, we looked at the following forest types: maple with an understory of Solomon's seal, summer oak coppice with a varying understory and a few birch trees, and summer oak forest with an understory of bracken. At the Oostereng, we sampled in Douglas forest with regeneration of Douglas, Norway spruce forest with blueberry understory, and Japanese larch with an understory of wavy hairgrass, regeneration of pine and shrubs. Ticks were collected using a white fabric of 1 square meter, which was dragged along a transect for 1 minute, which is equal to approximately 30 meters. The exact distance that we walked was measured afterwards. The ticks were then counted and put into pots using forceps and brushes. These ticks were also used for the lab experiment. Thickness of the litter layer and the density of the canopy were measured halfway the transect. The density of the canopy was measured using a densiometer, while the cover percentage of other vegetation layers was estimated by site. Per site, We made 3 random transects every morning and every afternoon for 2 days. In the lab, we created 10 miniature climate chambers with 8 satay skewers of different sizes. These sticks represented vegetation used for questing of the ticks. In each of the cups we placed 10 non-adult ticks. We chose non-adult ticks because these were the most abundant in the forest sites and therefore the most representative. Each of the cups received a different treatment. This table shows an overview of the 10 treatments. First, the 3 humidity treatments were kept at daylight and aimed for a humidity of 50, 70 and 90%. Secondly, we created 3 different light treatments, which were kept at 90% humidity in order for the ticks to be comfortable. Finally, we made 4 combinations of humidity and light to check for an interaction effect. For the dry treatment, we used 1 gram of silica gel. For the wet treatment, we used wet cotton wool. And for the medium humidity treatment, we used a slightly humid piece of paper. These items were put into a piece of black pantyhose. The relative humidity of the cups was measured using a humidity meter. The cups were then securely sealed using cling film and elastic bands to prevent the ticks from escaping. For the different light treatments, we placed the cups into an open box, a closed box, and a box covered with a green mesh. Light was turned on from 6 AM until 8:30 PM. The number of walking, questing, quiescent and dead ticks were counted every hour from 8:30 AM until 2:30 PM. After each measurement day, the ticks were removed from the cups. Each cup then randomly received a different treatment, after which new ticks were placed in each. And now to our results. We will start off with discussing the results of the field experiment. After sampling for 2 days, we obtained the following. This is a graph of the number of ticks per location. The x-axis depicts the site location. The y-axis shows the average number of ticks that we caught per drag. As you can see, the number of ticks varies among the different locations. Most ticks were caught in the larch and maple locations, while the other 4 locations revealed less ticks. But how can this variation be explained? Our hypothesis states that more ticks should be found in sites with a dense vegetation cover and a thick litter layer. To analyze if this is the case, we used the Poisson generalized linear model. We chose this model because the data shows a skewed distribution and contained a lot of zeros. Using a forward selection method, we included the most significant factors and covariates that might explain the variation in tick abundance. In the end, we included 3 significant drivers: litter layer, moss cover percentage and herb cover. We will start out with the effect of litter layer, which was added as a covariate to the model. In the table you can see that litter layer has a significant effect. Also, the p-value shows that this effect is positive. On the x-axis of the graph, you see the thickness of the litter layer in centimeters, while the number of ticks caught in a 30-meter drag are shown on the y-axis. This graph seems to correspond with the model in showing an increase in tick abundance with increasing litter layer. In our graph depicting herb cover, there seems to be a hump-shaped curve and not a linear relation. This is not surprising, as optimum curves are common in ecology. For this reason, we included a squared covariate of herb cover to the model. The moss cover percentage was added as a factor to the model. It was split into 2 values, low and high cover. The high moss cover was used as a reference and set to 0. The value shown in the table is the difference between the low and high moss cover. A negative value therefore means that there is a lower abundance in sites with low moss cover. Including tree cover to the model reveals that it is indeed not significant. The same thing is true for shrub cover. Having obtained the results, we invite you to evaluate them critically with us and put them into a broader perspective. First of all, it could be possible that 2 factors or covariates explain the same variation in tick abundance. For example, sites with high tree density also have a high moss cover. A collinearity analysis showed that all variables except litter layer are sensitive to this collinearity. To see which factors are related to each other, we also did a Pearson correlation analysis. The resulting table shows that indeed most of the variables interact. For example, forest sites with a high tree cover also have high herb and shrub cover and a thick litter layer. The implication for our model is that it shows significant effects, while in truth these effects exist because the correlated variables reinforce each other. As a result, only litter layer seems to have an effect on its own. Sampling in vegetation with bracken is quite problematic, as ticks are often found on the underside of the leaves. Therefore, the number of ticks has a chance of being an underestimation. Other studies avoid this problem by not sampling bracken vegetation at all. Another source of sampling bias was the avoidance of brambles and high vegetation during dragging. The fabric used for sampling simply could not be dragged through rough or thorny vegetation. We also found interesting differences between the sampling day, the researcher who did the sampling, and the time at which the sampling was done. The first day of sampling yielded less ticks. Also, more ticks were caught in the morning compared to the afternoon. Since the number of ticks caught corresponds to the number of questing ticks, this difference can be explained by more favorable questing conditions in the morning. For example, in the morning the humidity is higher. Unfortunately, we did not have the right materials to measure the exact microclimatic conditions in the field experiment. To test the significant differences of the lab results, we first calculated the percentage of ticks per behavioral activity: questing, walking, quiescence and mortality. After that, we tested for significance by using a generalized linear model. Statistics show that behavior is significantly influenced by light and humidity conditions. To illustrate the similarities, bar diagrams were made of the different behaviors at different light and humidity conditions and combined treatments. First, let's talk about light. Percentage of ticks is shown on the y-axis and light condition on the x-axis. All behavior types differ significantly per treatment, except walking in dark and light conditions. As seen in the bar diagrams, darkness has a positive effect on questing and walking, whereas light has a negative influence. Quiescence and mortality, on the other hand, are highest in light. Thus, it can be concluded that ticks prefer shady and dark conditions. But perhaps not only light has an influence on the behavior of ticks. Let's have a look at humidity. Also, with humidity, the differences are significant, but, like light, walking behavior does not significantly differ between dry and wet conditions. As you can see in the bar diagrams, ticks are more prone to be active in wetter conditions. Questing behavior is even the highest in the most wet condition. On the other hand, in the dry condition, mortality is the highest. This shows that ticks need a humid habitat in order to survive. In dry habitats, desiccation is a big risk. Now let's see what's the most important factor that influences behavior. To test this, we looked at 4 different combinations: dry and dark, dry and light, wet and light, and wet and dark. Here, all the dissimilarities were significant. As can be seen in the diagram for questing behavior, humidity has a bigger influence than light. Also, mortality is more influenced by humidity than by light. How drier the condition, how higher the mortality rate is. The differences between the light conditions is less dramatic. From these results we can conclude that humidity is the most determining factor for behavior of ticks. Light also influences them, but on a lower level. It can also be concluded that ticks are more prone to questing in a darker and more humid habitat, but dry conditions could be fatal for them. There are some improvements that we would suggest for future studies. First of all, our climate chambers were not very advanced. Thus, we could not very accurately adapt the conditions, especially for humidity. On top of this, we observed that many ticks started walking when we picked up the cups in order to count them. As a result, the number of questing and quiescent ticks could be underestimated. It would be better to develop a way of counting without having to move the cups. Having studied the results, what can we conclude from them, and do our hypothesis and prediction agree with this result? In the field, we saw that tick abundance varied per location. We were able to explain some of this variation by the difference in litter layer. The abundance of ticks seemed to increase with an increasing litter layer thickness. This is also what we predicted, so our hypothesis was confirmed. Furthermore, there seemed to be some effect of vegetation structure on tick abundance. However, we could not determine which vegetation layer was the most relevant, since vegetation layers appear to be correlated to each other. Our prediction was that the abundance would increase with denser vegetation in all layers, but because of the correlations, we cannot confirm our hypothesis with certainty. In the lab experiment we looked at light and humidity. We predicted that tick activity, and namely questing, is highest in darkness. From the results we learned that questing behavior was indeed highest in darkness. On top of this, ticks walked more in the shade. Thus, we can conclude that ticks prefer shady and dark conditions, which agrees with our prediction. For humidity, we predicted that tick activity mainly occurs in a more humid habitat. From the results, we can see that ticks quested most in the most wet condition, although for walking, it did not really matter if it was dry or wet. However, the mortality rate was the highest in the dry condition. From this, we can conclude that ticks need humid conditions to quest, whereas dry conditions can prove to be fatal. This agrees with our prediction. When we predicted the effect of interaction between humidity and light, we stated that ticks would more likely be questing in darkness with a high humidity. In the results we can see that this is indeed the case. Ticks quested more in darkest and wettest conditions. Also, you can see in the bar diagrams, the differences between humidity conditions is higher than the differences between light conditions. Therefore, we can conclude that humidity is the most determining factor for the behavior of ticks. When we combine field and lab results, we see that a thick litter layer can be a factor providing those humid and dark conditions that ticks prefer. So next time you're taking a picnic, think twice about where to sit down, or your picnic might just turn into a ticknic.